Well, let me thank you for joining this webinar. I'm Olivier Pacat. I'm head of scientific and technical development for Safic-Alcan. As you probably know, the IFSCC Congress is one of the most important yearly scientific event in the cosmetic world. And this year it was held in Cannes in France, and Symrise was awarded 5 posters. And we thought it would be a good idea to ask the authors to present the posters in 10 minutes for those who missed the IFSCC Congress. So let me thank you for participating. Let me thank the Symrise speakers for being there. If you have questions, you can write down your questions in the chat box, and they will be. We'll try to answer all of them. If not, we'll be able to catch up with you later. We'll send you the answer of your questions. We'll not be answering commercial topics this time, but only focusing on scientific questions. So, well, again, thank you for participating. Thank you to Symrise for their support. I will leave the floor to our next. Can you put the speakers, maybe, Morjan? Next slide. Okay. So you see, we'll have 5 speakers: Ricarda, Camila, Rebecca, Stefan, and Maria. Let me leave the floor to Ricarda. Maybe Ricarda, introduce yourself briefly, and she will be presenting a presentation about plant-based multifunctional. Ricarda, the floor is yours.
Thank you, Olivier. So my name is Ricarda Kreling. I joined Symrise 2016 and working now as a manager for functional efficacy applications. So everything that has to do with stability test with in vivo formulations. And before I worked at Henkel and Evonik. So let me start to share my screen. Changing to full screen. So, okay, I will give you a short introduction and going then to the multifunctional efficacy of our new Mindera ingredients. So as you all know, the product protection is essential to ensure the safety of a cosmetic formulation. And there's, of course, a growing demand for plant-based ingredient compared to the synthetic.
Ricarda, if you can just make that in full screen, I think we just have an issue with the screen sharing.
I will share again.
Yes, please. Thank you.
Better now?
It's not in full screen.
Again, not?
No. If you can make sure that like the screen that is shared is the full screen mode.
I know.
Yeah.
No?
No.
I will stop.
Yeah. Otherwise, we will just. Yes.
Working?
Perfect. Thank you.
I don't know what was no different. So, so as you know, there's a growing demand for plant-based ingredients, but chemical stability, the order, and also the efficacy is a very big hurdle for this ingredient group. We now developed an 100% plant-based solution, offering for product protection, but also offering multifunctional benefits. I will go into detail for this now. The first thing we evaluated was the chelating efficacy. Therefore, we checked for cupric and ferrous ion chelating capacity comparing to EDTA. And especially for maltol, which is one of our ingredients, you can see that the performance is really comparable to EDTA. So we tested 100 ppm, 250 and 500 ppm, and same concentration for the EDTA. And this is really comparable. And by binding these metal ions, there are different positive effects. There's also the availability of the nutrients for microorganism restricted. This is why this is really supporting also in preservative efficacy tests, but also supporting in general in your formulation stability. And we also tested this for ferrous ion. Maltol is not working so well compared to EDTA there, but still has a very good efficacy. And also anisic acid, which is one of the other candidates. The most important thing, if you are talking also about multifunctionals or preservative boosters, is of course the challenge test. And what is really obvious is that alone the ingredients do not lead to convincing results. So you can see, especially for Aspergillus, they are not working alone, but in combination, we are able to reach criteria A for pharma and ISO criteria. So this is why it's really, they are supporting each other due to the mechanism. What we also checked is, for example, the influence on emulsion properties. Therefore, we prepared simple oil and water emulsions, checking on the one hand the oil droplet size measured by Malpal master sizer, and there you can see a reduction of the oil droplet size, also reflected by the microscopic images. And the more homogeneous and smaller oil droplets also leads to an improved stability, which is reflected by these instability index, which was calculated with the Lumicizer and later with the DebView software. The higher the instability index, the less stable is the formulation, and you can really see that there's a strong improvement due to maltol. Okay. Then what we also did is really test antioxidant properties and the support of soothing. Here we have the results of maltol. You can see that the antioxidant capacity with 2.5 ppm is really comparable to the reference. And it's also dose dependent, and the same for Cinnamol. For soothing, it's also seen that the effect is available for Cinnamol and maltol, even at a lower concentration compared to the ginger extract. So coming to an end, trying to be time efficient. So you can see that it's a 100% plant-based solution offering full product protection with multifunctional benefits when combining them. For Mindera MA, which is maltol, we have a superior chelating capacity. There's an synergistic antimicrobial efficacy for Mindera maltol, syn, and anisic acid. The co-molding behavior of Mindera maltol improves the emulsion stability, and we can see that the antioxidant properties of Mindera maltol and syn are comparable to the reference. And just for teaser, so this is the start of a new platform with further ingredients to come. Thank you.
Thank you, Ricarda. So now we can move to Camilla's presentation. So Camilla will give you the end to present.
Can you see my screening, my
Yes,
presentation? Can you see them moving? Yeah, the
yes.
switchover. Great.
Thank you.
Okay, so good morning, everyone. I'm Camila Quesada from QTech, the Symrise Screening Services. I joined the team around 2 years ago, and today I'm going to show you some of the highlights of our project that we titled Ex Vivo Human Skin Model for the evaluation of rinse-off products. As you might know, rinse-off products can reach a penetration marker of up to 95%, and even though they are designed to be immediately washed off after their use, to a certain extent, some product might remain or be absorbed by the skin. So to test the assessment of their effects, whether these are beneficial or detrimental, is a key step before their market release. Unfortunately, the in vitro models, which are the ones mainly used for the testing of this type of products, present different challenges, mainly related to their lack of heterogeneity, and also they are missing the skin protein lipid barrier, and usually they are not strong enough to withstand the mechanical damage that can be um, um, related to the physical activity of rinsing of a product. However, we did realize that our ex vivo skin model overcome several of these challenges, as our model presents almost all the skin components, granting a correct skin metabolism. Also, it preserves the barrier function and its permeability, while the epidermis and dermis strength prevent this mechanical damage that, as I mentioned, it is usually related to the physical act of rinsing of a product. So this way, our main goal was to design an ex vivo model that allowed us to efficiently test or evaluate rinse-off formulations. And to this purpose, we established an experimental setup in which we use our ex vivo human skin explant, and we cultivated and treated them daily until reaching the experimental endpoint. The critical point of our protocols is that we perform treatments for a short amount of time, and then we perform a thorough wash or rinse off of the product, which is not possible to do in several of the available models to test this type of products. And then it is similar to what we should do or what we do with this type of product in the real life. So then after we evaluated different markers, in particular, we evaluated reactive oxygen species and skin barrier markers through image analysis. And our first point was our proof of concept of the model, to which we evaluated skin barrier markers using an standard rinse of formulation. This was an standard shampoo formulation without any active ingredient added. And we found a strong, we found a partial reduction of 2 out of the 3 skin barrier markers, which were desmoglein and Claudin 1. And even this might seem like a bad result or a bad indicator, is quite the opposite, as in literature it's been extensively described that shampoo formulations or rinse-off formulations in general might have a mild effect over our skin barrier. So these result points that our ex vivo skin model show responsiveness while maintaining its integrity, as you can see here, and the morphology. So we then pass to our second part of the proof of concept, which was related to the evaluation of an active ingredient. So this time in our shampoo formulation, we added vitamin E, which is a well-known antioxidant. And what we did is that we induce oxidative damage through the UVA light, and we observe a strong decrease of the oxidative damage through the measure of reactive oxygen species after the treatment of this shampoo formulation with vitamin A. These results not only confirm the first one, meaning we are confirming the responsiveness of the model, but also confirm to us that we were able to measure efficacy of active ingredients, not only observing standard formulations. So once we finished our proof of concept, we decided to evaluate two of our active ingredients of Symrise portfolio. The first one is a Symferment, a postbiotic derived from biofermentation of selected lactobacillus strains. And we applied the same rinse of protocols, and as you can see here, we found a strong promotion of Claudin 1, desmoglein 1, and filaggrin, which are again three skin barrier markers which are pointed to a strong barrier reinforcing effect of Symferment. Which is very important to notice is that even though you are seeing now 30% of Symferment, our protocol involved the dilution to a 10%, so a 10% of this concentration before the applying. So in reality, these results were obtained with a 3% of this product. Plus, this was only 15 minutes treatment daily for 3 days. So we are seeing not only a good responsiveness of our model, but a great supporting skin barrier effect by this active. So then we move to our second active ingredient, which is Symreboot L19, a Cosmos-approved paraprobiotic active. And again, we observe a great and a strong promotion of Claudin 1, desmoglein 1, and filaggrin. Again, I think it's important to notice that we use the same protocol. So in shampoo, we diluted to a 10%. So it's in reality what we were using is 0.1 and 1 and 0.5%, sorry. And the application was only 15 minutes treatment daily for 3 days, which again points a clear results for our model, but also the great skin barrier reinforcing effect of Symreboot L19. So finalizing, we found that the data presented in this work established and validated our rinse of model as a tool for screening rinse of formulation that, as I mentioned in the beginning, has presented several challenges until this date. Demonstrated that our model present responsiveness with measurable changes in key barrier markers, for example, Claudin 1, desmoglein, and filaggrin. Also that our model present suitability for more complex analysis, as for example, we are also to evaluate reactive oxygen species damaged, which is a more complex setup protocol, while the model maintains its structural integrity throughout the evaluation that, as I said before, is not observed in all the available models, mainly in vitro models. And also our model showed suitability for the evaluation of active and more complex compounds and formulation, as we saw with Symferment and Symreboot, respectively a postbiotic and a paraprobiotic in shampoo formulation. So thank you very much for your attention.
Thank you, Camila. So now we can move to the next presentation, which is Rebecca. So Rebecca, you should now have the end to present. Thank you. It's perfect. It was
Okay.
working well,
Yeah.
Rebecca.
Okay. So you can hear me now. So hello, good morning. I'm Rebecca Gasparin. I'm the Hair Care Center manager at Symrise, and I'm working for Symrise for 10 years now, and I'm a pharmacist, and I have a master degree in pharmaceutical sciences, and I'm finalizing my PhD also in pharmaceutical science. And today I will show you some of the new results we have for our 360 degrees platform for hair reconstruction using green ingredients. So first, I would like to talk a little bit about hair. So hair is a keratinous appendix, and the hair fibers grow from the hair follicles that are located in the skin and scalp. And when the hair fiber first emerges from this scalp, it's totally keratinized, so we consider it as dead tissue, meaning that it's not anymore live cells, or it doesn't have metabolism, active metabolism. About the hair fiber, we can divide it in four main structures. So the outermost layer is the cuticle. It is made of 5 to 10 layers of flat and overlapped cells. And above, so this proteic cells, we have the F layer, that is a lipid layer, mainly composed of 18 MEA. And the cuticle is mainly the main function of the cuticle is the appearance of the hair, the protection against external stressors, and also it's important for the diffusion of active ingredients, for example, from the external part to the internal part of the hair fiber. The second structure, that's the main one when we think about mass or size, is the cortex. It's mainly formed by fibrils, so keratin fibers, surrounded by a also proteic matrix. It's important for the strength of the hair, so all the mechanical properties of the hair are related to the cortex, and it's also in the cortex where we found the melanin, that is the responsible for the hair color. The third structure is the cell membrane complex, or CMC. It acts like a cement or a glue that connects all the cuticle layers between themselves, and also the cuticles with the cortex. So it's important for the cohesion of the fiber, and also it's important for the diffusion, because some ingredients can pass through the CMC, so between one cuticle layers and another and the other. And the last one is the medulla, that is the inner part of the hair fiber. It's a porous and intermittent structure, so it can be present or not in the hair fibers, and it's important, for example, for thermal insulation. So when the fibers first emerge from the scalp, we have all these structures really healthy. So we have healthy hair. They are totally well structured, but then we leave. So the life damages the hair. So we exposed our hairs to sunlight, to pollution. So the hair gets dirt, and we have to wash it using like detergents, for example, strong surfactants, and then we will comb the hair to detangle it. We use heat styling tools to shape the hair in a different way, or to speed up the blow, the drying process. We also want to change the hair color or shape using really strong chemical treatments, or also again heat styling tools. We can braid the hair. So all of these procedures, let's say, damage the hair in different ways, and this damage will impact hair structure, physical chemical properties, and also the overall behavior of these fibers. And the consumer will perceive it like the hair is more prone to breakage, it lost the malleability and shine, we have more frizz and roughness. So for the consumer, it's a really important subject, like hair damage is considered one hair type, and it's one of the three global hair concerns for 54% of the consumers. Also, when you look at social media or like Google Trends, we saw that how to recover damaged hair has search for how to recover damaged hair increased like 100%, and damaged hair is among the most searched terms in the hair category on Google Trends. Also, in the TikTok, we saw a lot of search for hair damage, and also about, and more important, to how to recover damaged hair. So thinking about that, we normally have two different strategies to treat the different hair layers. So when you're thinking about the hair surface, we are talking more about smoothness, malleability, and like chemical and physical barrier. And for that, we can use materials like larger molecules that will deposit on the hair surface, like emollients, lubricants that will condition barrier this external part of the hair, like positively charged or cationic materials that will also have better substantivity in the hair, because we have, when we damage the hair, we have more negative charges. And one example of one of the materials that we can use is protein. When you think about the cortex, it's more related to nutrition, resistance, strength, and mass restoration. And for that, we can use materials like small molecules that will be able to really penetrate into the hair fiber. It's important to have a good polarity, like dipoles and shards that will again take that, like have the ability.
I think we have a small freeze. We'll just wait for Rebecca to join back.
Rebecca, yeah.
Okay.
That's in product.
Oh, sorry,
Sorry.
Rebecca, we missed you for a few seconds. If you can maybe just finish the over slide.
Previous
This one?
slide. Yes.
Yeah,
Yeah.
this one exactly. Thank you.
Okay, so we missed the cortex part.
Yes.
Okay, so for the cortex, it's important that the desired benefits are more related to nutrition, resistance, strength, and mass restoration. So it's important that the ingredients are able to really enter the hair structure and reach the cortex. So for that, we need small molecules that has good polarity, like we need some dipoles or charts that will make the ingredients able to really interact with the damaged sites in the hair. And some examples of materials that can do that are organic acids and peptides. So another aspect that is really important is the greenification of hair care. So it's important that we have green and sustainable ingredients that are not only good for your hair, but also good for the planet. And at Symrise, we work with this 360 approach for hair repair, considering ingredients selected from nature that have specific actions based on its affinity for the different hair structures and also the size. And we thinking about some combinations of ingredients to have in the end a synergistic effect. So we selected the first three ingredients from our portfolio. So the first one is in Hair Restore. It's a high molecular weight wheat protein. It's native gliadin. We say it's a smart protein because it actively binds to the most damaged areas in the hair, and it can provide like film formation, improve the hair condition, split ends repair, and anti-breakage effect. The second one is in hair shape and color. That's a lipid-rich composition with wheat extract, camellia oil, and also vitamin E. It works as a CMC replacement, improves hair fiber cohesion and overall health, providing cuticle alignment, color retention, volume freeze control, and also curl retention and definition. And the last one is synprotein lupin. That's a biotech-derived low molecular weight peptide from organic lupin, and it works as a bond builder, so it penetrates deeply in the hair cortex, establishing new bonds and interactions and reinforcing the hair structure. Provides nutrition, mass restoration, elasticity, balance, increased resistance, and anti-breakage. So you can see that we have different solutions for different parts, let's say, of the hair. And so we thought about what can happen if we put them together. And the first combination was Syn Hair Restore with Syn Protein Lupin. So a big protein with the small peptides. And here you can see the results we have in the fatigue test. The fatigue test is an assessment that can where we can have a survival probability graph, let's say. So we pull the hair fiber a lot of times, and we register the number of cycles that this fiber can resist. And it's really connected with the daily grooming. So when we comb the hair, when we tie the hair, when we braid the hair, for example, and we create these small fractures in the hair fiber that will propagate when we again do these little stresses to the hair. So here you can see that we have the placebo group, placebo-treated groups with shampoo and conditioner without the active ingredients. And when we add like synprotein lupin at 2%, we have better results. But when we add like synprotein lupin at 1%, so lower dosage with 0.5% of Syn Hair Restore, we have even better results. So when we look at the characteristic life, synprotein lupin alone increased the characteristic life in 27%, and the combination in lower doses increased the characteristic life in 39%. And the second combination that we studied was syn hair shape and color with synprotein, so a CMC replacer with a bond builder. And we studied the effect by the repeated grooming assessment, where we have like an automated combing machine that will comb the hair a lot of times, and we can collect the fragments that are generated by the hair breakage, and we can count the number of fragments. So here we can see that the placebo formulation have a higher amount of fragments, and then with synprotein lupin we have a 28% reduction, with shape and color alone 44%, and with the combination we have 76% reduction. So
we lost the speaker again.
I don't know if we lost
Okay.
or maybe we can
Go to the next one.
move.
And then,
so sorry.
Yeah, if you can just like finish, Rebecca.
Yes, it's
because we
the
just need
last one.
your end. Thanks.
Okay, so we achieved synergistic effect combining the green technology with different mechanisms of action, and so it's an important study to show how natural, sustainable, and biodegradable materials can offer innovative solutions for complete rare reconstruction.
okay, thank you.
Okay.
For the next
Thank
speakers,
you.
if we could
Yeah.
keep the 10 minutes so that we can have some
Sorry.
questions. Please.
Yes.
Thank you.
So we'll keep going with the hair care, and now we will move to Stephen. Stephen, you should have the end on the presentation now. Thanks.
No, you're on mute.
It works well. Oh yeah. Perfect. Thank you.
Sorry for that. So I have to share again. So you've now a perfectly preserved formula. We have smooth skin and nice hair, but there is still dandruff available. So let's cover that topic as well. So first of all, a little introduction about dandruff. So there is two kinds of in the end of dandruff. One is due to high sebum content, and the other one is to dry scalp and to low sebum content. So we like to be in the middle of that for that and focus on the real dandruff here. So we have oily scalp and hair. We have very big flakes that are sticking to the scalp. It's itching, it's red, and everything's bad. So let's treat that. And for that, we have an ingredient on the market which is perfectly known. This is pyroxene olamine. It has very good efficacy. This is in vitro data against different Malassezia species, and again against Staphylococcus. And we see in vivo studies that it works perfectly fine. So if you treat your scalp, you get less dandruff, and it works perfectly fine. As usual, if we have something on the market that it works kind of fine, well, our colleagues from sales ask us, how can we make that even better? Because the pyroxene olamine, it's a powder. Well, it's hard to formulate. So we had a look at our portfolio in dandruff related ingredients. We have quite some established ingredients like ciclimbazole and the pyroxene olamine, which are on the market since longer days. We have modern solutions like propyndole caprylate, propyndole andesylonate, and desylene glycol. And we have already one liquid blend on the market. So we try to get this all together to come up with a new blend, which is, well, kind of has the same efficacy, which is liquid, more biodegradable, and not that much more expensive. So like a task that is not that easy to solve. And therefore, we tried combining again different ingredients that we already have. So Paraquinolamine was central because we like its efficacy in vivo and in vitro, and we added here. So we came up with adding diethylene glycol, 1,2-hexanediol, and glycerine. All those four ingredients participating in the final formulation that has kind of the same efficacy as we saw already before for paraquinolamine, but with added values through the synergistic features that all the ingredients bring along. And first of all, we tested this in vitro, so on Malassezia furfur and Sympodialis. And here you can see the paraquinolamine. This is a small round here, and Cicrinpan PO+, which has a broader range here. So what you can see, all these agar plates are covered with these fungus, and where the larger this circle here is, the more efficacy you get from an ingredient. So you can already see here through one picture that it's even better than paraquinolamine alone. This is on the graph to display this, and the less, the more, the better here. So, and you can see this in numbers as well. So in zone of inhibition, it's already better than using paraquinolamine alone. And here you can see the answer to the biodegradability question. So the paraquinolamine still is not biodegradable, and we cannot do magic to make it that way. So we added ingredients like hexanediol, diethylene glycol, and glycerol that are readily biodegradable, and so we got into 95% of ingredients that are already readily biodegradable without losing efficacy. Then, so we come to an optimized performance against Malassezia, and we were able to produce a real liquid blend. So you won't need any hard steering in the process, in the formulation, and you would not heat to solubilize the ingredients. So this will add to the performance of this ingredient. But the important question is, does it work? And the answer is luckily yes. So here you see the direct comparison between PO alone and PO plus here in dark blue. And you can see two things here. One thing that was already expected from the in vitro data that I showed you, that PO plus is more effective than PO alone. But there is a second cool thing, and this is, while you lose efficacy after stopping the treatment here with the PO, you keep the efficacy here even through the washout phase, at least for 14 days that we checked. So you have here a long-term solution for dandruff. And of course, we have subjects to display this exactly what happens. And already here, you can see some differences between the photos that we have through on the scalp and the outcombed Malassezia or scalp things here that we find on the black spot. We have also an improvement in itching, which is very important when talking about dandruff. And we can add to our further experiments that we can improve the scalp barrier function here. So all this contributes to a better feeling of your scalp and the reduction of dandruff. But there's more. You might realize that we add 1,2-hexanediol in this formula. And this helps us also to get a better, to a support of the product protection system. So you can see here the the challenge test results that, well, the format that Ricardo already showed you, we used here 1% of PO plus with potassium sorbate in different formulations to assess the efficacy here. And we were already with these two ingredients, we were quite successful. Now imagine if you added all the things that Ricardo showed, that would be, well, a nice effect. To combine this, we have used the synergy of every ingredient we tested, and every component takes part in that. So and adds a function, and we ended up in an optimized performance against Simalacetia. We have even faster performance than Peractinolamine and a long-term effect, and we have an easy to use liquid, and it even supports the product protection system. So in the end, this is more than what was asked of that, and we were quite proud to deliver such a system. So thank you for your attention.
Thank you, Stefan. So now we can end with Maria. So Maria, you're now the presenter.
Hello. So I share my screen, and I guess you see now the presentation. My name is Maria. I'm a biochemist by training. In the past, I did a lot of cell biology on skin cells, and I'm with Symrise since 2019 as a lab manager, where we develop actives for skin biology, and we test here all our ingredients on certain efficacy, for example, inflammation. And today, after the nice talk of Stefan about anti-dandruff ingredients, I would like to present another active ingredient for itchy skin, which is Safic-Alcan Avena. And for this ingredient, we performed nice mode of action studies on a new receptor, which is essential in mast cell regulation, which is called the mast-related G protein-coupled receptor X2. And why is it so important to develop soothing ingredients? So for a customer or consumer insight study, we know that global consumers think that they have sensitive skin and that their sensitivity increases over time. So 73% of global consumers think that their sensitivity increases, and 36% of the global consumers perceive their sensitivity as an itchy skin. And that's why we developed a hero ingredient from oat. So oat is traditionally known to exhibit efficacy on inflammation since lots of years. So there's a traditional phytomedicine. And for example, in US or in Germany, it has been long known for its itchy and anti-itchy and anti-irritation efficacy. And here we developed a nice ingredient from organic oat grains by a new technology called SymTrap. That means we perform an oat extraction and then enrich the active ingredients from the oat by desorption absorption technology with a resin that enriches the positive molecules, the useful molecules, and thereby recycling the byproducts. And thereby we enrich the avenanthramides. The avenanthramides are the active ingredients in oat. And here we have a standardized extract which contains 200 to 250 ppm of avenanthramides, which we then spray dry and have a nice solid ingredient that can be very well handled. And in order to explain you how our Safic-Alcan Avena works, I would like first to present you the pathway of the itchy skin. So in the skin, you have exogenous triggers like microbes, pollution, or even UV irradiation, which all act on the nerve endings or the keratinocyte, thereby inducing an inflammatory cascade. And here mainly it is to say that substance P is in this cascade produced, and substance P acts on the mast cells, thereby activating the mast cells. These degranulate their vesicles and histamine is released, and this histamine then can act again on the nerve endings and mediate pruritus, so itching sensation, and this also mediates redness. In our case, the Symcalmin Avena acts on the activated mast cell, namely on the MAS-related G protein coupled receptor and also the NK1 receptor, which both are able to bind substance P, and Symcalmin Avena is able to modulate this activation and thereby also modulate the mast cell degranulation and histamine release, and thereby soothes the pruritus sensation and also the redness sensation. And in the next, I would like to explain you how the avenantramides and the oat extract is doing this. And therefore, I would like to introduce you the pathway of the MAS-related receptor. And first, the substance P binds to the receptor, and this receptor becomes activated, and thereby a calcium influx into the cell occurs, and ERK, which is a MAP kinase, becomes phosphorylated. And these two signals then lead to the mast cell degranulation. That means that all these vesicles, which are stored in the mast cell, fuse with the membrane and release their cargo to the outer space, and thereby the itch sensation is mediated. And the avenantramides then are able at the level of the receptor to inhibit the activation of the calcium and the phospho-ERK, and this is what I would like to explain you with our studies. So we first performed a receptor study on the MAS-related receptor, and this was done in an reporter cell line. This reporter cell line expresses the MAS-related receptor together with a beta-arrestin construct, which together with the receptor in presence of substance P fuses, and the receptor is linked to an enzyme, and this enzyme is completed upon activation. That means these two donor and acceptor fragments fuse, and in the activation state, beta-gal can be active, and the substrate is catalyzed to a light signal. And with this, we are able to monitor the activity of this receptor. And when we add substance P to a certain dose, we can see that there is an activation dose dependently, and we see that this construct is working. In the presence of our ingredients, the oat extract, we can see that in a dose-dependent manner, the substance P activation is inhibited, and therewith we can already say that the oat extract at the level of the receptor inhibits the mast cell degranulation. And we show this in further studies. So next, we also looked at the calcium influx, and we have seen by cell flow cytometry that upon stimulation of these cells, here in the untreated control, there is an increase of calcium in the cells. And when we have the oat extract present, we have a reduced calcium influx in the cells, and thereby seeing already that there is a lower level of mediation of the receptor activation. Exactly so. Safic-Alcan Avena decreases the calcium influx in the mast cells. In the second signaling, we also looked at the ERK phosphorylation by Western blotting, and we see here on the right-hand side that the ERK phosphorylation in the presence of oat extract is reduced with 5 micromolar and 10 micromolar. In the absence of the oat extract, we see a strong ERK phosphorylation. And with this, we can also say that the oat extract, the Safic-Alcan Avena, reduces the ERK phosphorylation after activation of the mast cell receptor with substance P. And finally, we looked at the mast cell degranulation. The mast cell degranulation can be monitored in 2 ways. First, we can measure the beta-hexosaminidase release. This is an enzyme which is which facilitates the inactivation of microbial peptides, for example, or microbial triggers. And second, we can also monitor the degranulation by receptors that are intracellularly in the vesicles, and by vesicle fusion, which is the degranulation, these receptors which are intracellularly become exposed on the mast cell membrane, the surface, and this surface expression can be monitored with an antibody in flow cytometry, and we performed both of those experiments. So here you see the data for the beta-hexosaminidase release. So when the mast cells are treated with substance P, you see a nice increase. So the hexosaminidase is released, and with the extract present, we decrease this release. And further, you can see that the CD63 and LAMP1 receptors are increased tremendously with substance P, and also other triggers we have seen and also tested, and the oat extract always tremendously decreases this induction. And finally, we also wanted to know if the Safic-Alcan Avena and this signaling that is inhibited by our oat extract also is able to reduce itchiness, and therefore we performed an in vivo study with Safic-Alcan Avena, which is a prick test. That means that the volunteers become pricked with histamine to induce an allergic, similar allergic reaction, and we see that the itchiness is significantly reduced by Safic-Alcan Avena, and there is together compared to a positive control in antihistamine, a very good efficacy. And this is an instant itching relief. We also performed an in vivo study for longer term, and here we measured a discomfort sensation by a questionnaire, and by averaging these questionnaire and analyzing the questionnaire, we found that the discomfort sensation scoring reduces the discomfort in the patients, and thereby we can also say that over 2 weeks there is a significant reduction in itching, burning, heating, and tightness stinging sensations in all the patients. And with this, I would like to summarize. We have here a nice ingredient from oat that is in a powder form with standardized levels of enanthromides stably in the ingredient, and we have now identified a nice mode of action on the mast cell-related mast-related receptor in mast cells to really know how the ingredient is performing, the itch reduction, and the soothing efficacy. Thank you for listening, and yeah.
Thank you very much to all of you. So I know we're quite short now on time, but we will just take 2 questions because I think they're like super interesting. The first one for Ricarda about the Mindera. If you can tell us a bit more about the biodegradability of the ingredients.
Yeah, all ingredients were tested and they are already biodegradable. So also a good opportunity compared to the synthetic ones.
Perfect, thank you. And maybe one last question for Stefan. So how many blends have you checked before coming to this nice composition?
Yeah, to be honest, we tried using AI here. However, it was not successful as the parameters were too high. So we used scientific intelligence instead of artificial one. And so we had around 40 different ingredients, and we have 4 different blend parts. So you can do the math by yourself. It's a 3-digit number, yeah.
Yeah, great. Okay, so I think we'll stop now with the question. I saw also some question about more like technical things and about the presentation. So everything will be sent to you right after the webinar, along with this recording video if you want to share with your colleagues. I will leave you, Olivier, finish and give it the final
Yes.
word.
Well, let me thank the Symrise team. I think that we had some very nice scientific work. Let me thank you for that. Again, sorry we cannot answer all the questions, but we'll be sending you we'll be answering this question via email and send you the answers there. So let me thank everyone for participating to this this webinar, and let me wish you a very good day to everyone. Thank you so much. And we'll have the recording of the session, and this recording of the session, as well as the presentation, will be available to everyone. Thank you.
Thank you. Have an excellent day, and wish
Bye-bye.
you
Thank you,
also.
Marcia, and also for
Bye-bye.
Thanks a
Bye.
lot.
for coordinating.
Thank you all. Bye-bye.
Bye-bye.